and allow me. That looks like nearly supernatural powers, right? You utter it and it occurs. Thank you very much, Esther. Shout out to Esther for helping pull all this together. My name is Ken Goodman. I direct our Institute for Bioethics and Health Policy at the University of Miami, and it is my pleasure and privilege to welcome you to the first episode in our annual series of dialogues in research ethics. Despite what the logo says, we're actually in year 31. And that means that we began this project, well, a really long time ago. This is the 205th dialogue in the series, where we've included faculty, visitors, and others talking very broadly about issues in science, in research, construed broadly. So we have here today 2 scientists. In the past, we've had lawyers, philosophers, clinicians. We had a clergyman do one once, as well as others. And what we hope to do in these dialogues is, well, inform our colleagues about issues that arise in the conduct of research, and also to foster discussion. So we call them dialogues in research ethics, not stupefying lectures in research ethics. I do enough of those, and I think people have heard enough of them. It is my pleasure today to welcome 2 colleagues at the University of Miami, Professor Dr. Helena Solo-Gabriele. As you can see from the flyer, she is at the University of Miami, Professor of Chemical Environmental Materials Engineering. And we have a special guest today, Professor Dr. Chris Mason, Professor of Computational Genomics in the Department of the Program of Computational Biomedicine in the Institute for Computational Biomedicine at Weill Cornell Medicine in New York. Helena and Chris are among the leaders of a very exciting NIH-funded project, RADx-RAD. RADx, for those of you who know it, stands for a Canadian channel in Category B owned by Blue Art Media. Oh, no, that's a different RADx. There actually is a RADx Canadian television program. The one we're talking about is the Rapid Acceleration of Diagnostics. And what Helena, Chris, and their colleagues, I see a number of other colleagues from that project. I see Cynthia Beaver. I think I saw George Grills a minute ago. Are leading a project to analyze wastewater, originally for COVID, and now for a potentially far broader number of maladies, but also biomarkers and signals that might be of use in everything from public health to oncology. Let me thank you all for being here, especially to our presenters and our guest, Professor Mason, who drove all the way from New York. No, sorry, that was a different visit. Thank you, Chris. I think Helena's going to get the ball going. She does have some slides, and with that, I'm going to shut up and hand the baton over to you. Thank you very much.
Thank you so much, Ken. Yes, this is a tag team presentation. I'll be presenting the first part, and Chris will be presenting the second. And I'm going to try to share my screen, but it says the whole host disabled participant screen sharing. So maybe Esther, can you help me out here?
I'm doing that, Helena, let me just shout out to our colleagues. This work is important for a number of different reasons. And so not only faculty are here, but it's a privilege to be able to welcome our colleagues from the Florida Department of Health and Miami-Dade Emergency Preparedness Consortium. We are participants in our communities. And with that, I think you're good to go, Helena.
Okay, and I think I've managed to share my screen. See my screen?
Yes.
Okay, and thank you so much, Ken. And of course, Ken is a member of our RADx-RAD team as well. And thank you for the opportunity to present. And the official title of our project is Development of Proof of Concept Implementation of South Florida Miami RADx. We are funded through the NIH, and our goal is to characterize outbreaks from measurements of wastewater. So we're combining wastewater measurements along with clinical information that's available. We have a data integration platform led by Stefan Schur. And then we put that all together, come up with prevalence rates of illness within the community. And we provide this information to decision makers who then make policy decisions based on the results from wastewater. And how does this all work? We know from the COVID-19 pandemic that transmission occurs from person to person through aerosol respiratory droplets. In addition to that, humans that are infected, whether or not they're symptomatic or asymptomatic, will release the organism through their urine and feces. And therefore, we have a repository now of those who are infected in the wastewater. So we can go to the wastewater system and then collect a water sample and then process that sample. And we have some brilliant molecular scientists who are able to take this information and extract the DNA, RNA signature from the SARS-CoV-2 from infected individuals. So overall, our ultimate objective of our project is to relate the wastewater to predict COVID-19 cases. So our study is designed such that we pair information available on the human health side with our wastewater monitoring. And the list of the human health information that we have available to us includes very significantly the elaborate testing, tracking, and tracing process at the University of Miami campus. We were the only school in South Florida to remain open, university in South Florida to remain open during the pandemic. And so we have this very rich data in terms of our student population, in terms of positive versus negative, and also amongst our staff. So one main wastewater group that we sampled from included all three campuses of the University of Miami, including the Gables campus, where we had about 20 locations set up, several locations at the medical campus, and then also at the Erasmus campus. So all three campuses were represented. In addition to that, we have a wealth of information from the hospital. The interesting thing about the hospital is that the patient population is well defined. We know what illnesses they have. And so we also made specific efforts to collect samples from the university hospital. We also have information from Miami-Dade County wastewater treatment plant. We had information from the FDOH on the prevalence of COVID-19 within the community. So we did sample, or continuing to sample weekly at the Central District Wastewater Treatment Plant. And then also effective January 2022, we collaborated with our colleague at the medical school, pediatrician Dr. Lisa Gwynn, who had a RADx Up project, very much related to the RADx RAD project that we are funded through, and joined forces and are continuing to collect samples of wastewater at Miami-Dade Public Schools. The majority of the schools are in Northeast Dade County, in addition to a school very close to the medical campus. And so now we have this wealth of information where we can match the information from the public health or the health information to wastewater information. We collect samples. Our sample collection process is pretty straightforward. This is the example of how we collect a grab sample. We also collect a composite using equipment that is capable of combining samples together. But essentially, it's a matter of opening up the lift station or the manhole, and then dropping some vessel to then collect the sample. And we illustrate here the process. The most important thing in terms of sample collection is to make sure we're safe. Those who are collecting the samples are safe. And then also that we protect the environment. So we have double containers in terms of, so we avoid the spillage of the wastewater once we bring it up out of the sewer. We have disinfection throughout the process to assure that we don't cause contamination of the environment and of ourselves. Once we collect the wastewater sample, that sample then is split amongst various laboratories, split three ways. It gets sent to the Center for AIDS Research here at the University of Miami, Dr. Mark Sharkey, where he runs an innovative technology called V2G, or Volcano Second Generation qPCR. In addition to that, the samples are sent to the Oncogenomics Shared Resource, also at the University of Miami Sylvester Comprehensive Cancer Center. And there it's analyzed by RT-qPCR by Sean Williams and Ben Corral's teams. Also at the Oncogenomics Shared Resource, deep sequencing process occurs where the samples are analyzed, processed for variants. The information on variants, the informatics is then processed at Weill Cornell Medicine, at Chris Mason's lab. And so we're able to generate reports from the qPCR data that we then send to University of Miami administration on a weekly basis, Miami-Dade Water and Sewer Department for the county data, and also Miami-Dade County Public Schools. So 3 reports get sent out every week. The data from the Oncogenomics Shared Resource, we cross-check it, the RT-qPCR with the V2G. on a weekly basis. And then at the Weill Cornell laboratory, they do a process called shotgun sequencing, so they can see all the microorganisms in the sample, and then also process the information from the deep sequencing using a bioinformatics approach to look at the SARS-CoV-2 variants. Just a quick update on some of the results. I wanted to share the campus data as illustrated here. We have clinical cases shown on the left axis, and then the bar graph corresponds to on-campus cases. We have both the students and faculty on a stacked bar graph illustrated in referencing the left axis. On the right axis, we have the log base 10 logarithm of the genomic copies of the virus that we see in the wastewater, the SARS-CoV-2, is given by the purple symbols. And what we see is by comparing the purple symbols to the case information, and again, the solid line here represents the 7-day moving average of the cases, so it's a lot easier to see the correspondence, but there is a strong relationship between what we see in the community and the levels we see in the wastewater. In addition to that, we have the vaccination information up at the top, and the gray line corresponds to employees' vaccines, and then the students shown on the red. And what we see is once the student population was vaccinated, a strong decline in the SARS-CoV-2 in the wastewater. There were several weeks where we were below detection limits, but then with the Delta wave, it resurged, and then with the Omicron, it resurged again. Also of interest is that the relationship on the Omicron data, we have a lot of people cases of illness in the community. The wastewater did go up, but not, in proportion. And so we have a very steep relationship between, this axis corresponds to the log of the wastewater to the cases per 10,000 people. A very steep curve, a little bit of an increase in the wastewater number resulted in a very large number of people becoming ill during the Omicron wave. And looking at hospitalizations, the correlations here are really, really, really good. The solid line corresponds to hospitalizations in the county as a whole, and we have the SARS-CoV-2 in the wastewater. Again, we have very strong correlations. What's very interesting in the hospital data, as opposed to case data, I showed you the case data for the campus. We also looked at the case data for Miami-Dade County as a whole, very similar to the campus data. But the hospitalization is different in the sense that the initial wave was the most steepest wave. So a little bit of increase in the wastewater. We saw a big increase in the hospitalizations. We saw that for the initial wave early on, for the Delta wave as well. And then when we hit Omicron, as opposed to the case data, just documenting the illness where we saw a very steep curve, when it came to hospitalizations on the Omicron wave, it's a much flatter curve shown here. So although we see a lot of people getting ill with COVID-19 during the Omicron wave, there's not as many hospitalizations. And then the post-Omicron, we're seeing elevated levels in the wastewater, as you can see by the green line here, and the numbers in the hospitals are very low. So the relationship between SARS-CoV-2 in the wastewater versus cases and hospitalizations, we've been observing them to vary over time. So at this point, I'd like to hand it over to Chris, who will describe more detail in terms of the shotgun sequencing and the deep sequencing that was conducted.
Yeah, so thank you, Dr. Solo-Gabriele. It was really it's been a pleasure to work on this project, and still ongoing, of course, and many variations of it. I think it'll keep going, if all goes well, far into the future. And this really changed, I think, how we view cities and municipalities and what we can measure. And it's actually where you don't have to ask the patients for samples. As we were just talking to the NIH yesterday, it gives you arguably the most unbiased view of what's moving and transmitting in a community. So that was a lot on SARS-CoV-2 and COVID, which was a great summary, and also comparison to some of the other hospitalization and biomedical data. But now I also want to look at other aspects and other organisms that are present that you can monitor in data that are relevant for human health, and basically other biology we can measure from these samples. So next slide, please. So what we've been doing is looking, you can kind of see that there was, there's Alpha, Delta, Omicron, different waves of the virus, of course, have come and gone Probably some of you in the audience are carrying maybe a newer strain of the virus as we speak, but they are now, we've now tracked many versions of them and there's new ones emerging. The way we do it, this is a very technical slide, but to walk you through is basically we align the reads, take fragments of RNA that get sequenced, map them to the known genome that was originally from Wuhan, the strain. And then you can see that reference, and we trim, basically clean up the file, clean up the reads, because you get all these, you get millions and millions of small molecules, and we basically trim them, clean them, map them to the Wuhan strain, and then look for differences, basically call variants, meaning we see, is there something in this molecule that we sequenced, which is the AC gene T, that's different from the reference strain, or actually there's a uracil in there because it's RNA, but you can see, the differences between the reference. And then we look at the mutations to see, well, which strain is it? Does it look like it would change the function of the virus potentially? And also look at the variant of concern lineage, or VOC, with a tool called Freya. And next slide, please. So that's what we use. Now, what we've seen with some of these samples, you can see is many, many reads. Again, we get sometimes millions of reads per sample. And you can see most of the fragments of RNA that we sequence does map out to the SARS-CoV-2 genome in pink. We do, however, see other things pop up. So you see the brown is you'll even get bits of bacteria, you'll get some occasionally fungi, archaea, some human RNA occasionally pops in there. Again, these are from human wastewater, so you'll see some human RNA that's floating around. So it's interesting to see there's really a broad ecology in the data from these samples. And then in this, each one of these vertical lines is just a single sample of all the samples we've sequenced so far with this protocol called Arctic, which targets the virus mostly. Next slide, please. And what we've been able to do with this is actually, cover the genome. This is from left to right, the whole genome of the virus. It's, basically about 30 kilobases or 30,000 bases long. And it has these different regions of the genome you've probably heard about. So like the spike protein you can see up there on the top is the area that actually interfaces with human cells and gets through the ACE2 receptor, and of course leads to the virus getting in, making copies and making you kind of snotty and coughy. And sometimes very sick, right? So that area we can see we get good coverage like we do other genes in the viral genome is what's listed on top. And the coverage means we're getting a lot of reads mapping there. So we can actually call mutations and characterize the virus for the ones that pass our filters on top, you can see. And other ones, we still get fragments of RNA. You can see where it starts to get splotchier, you get more blue. And that's because it is RNA that's moving through wastewater that of course has bits of fragments that can happen. It gets degraded. But what's amazing is that sometimes wastewater can be in there for days, maybe even weeks, and we still pick up fragments of the virus and you can still sequence it. So it's still there, even if it's a bit fragmented, you can still grab it and sequence it. Next slide, please. Then what we do then is as I described before with the method, we tracked all the mutations that have occurred basically since early on in the pandemic. You can see the alpha waves up at the top was kind of the blue. It's kind of hard to see the font, but you can see blue up top is alpha. And then we got into the delta wave. You can see kind of start to spike in, and then eventually Omicron, and then really Omicron. You can see anytime you see a red dot, that actually means a different mutation popping up in the genome. And you can see Omicron is carrying, as you move from left to right, it's over time, over the course of sampling for the past couple of years. We're getting more Omicron, and you can see in the bottom, at the top is mutations unique to each strain for each clade, like Omicron, Delta, Alpha. Those are the different types of it. And on the bottom are mutations that are shared across each of the kind of strains. So this is Alpha and Omicron. And you can see now the virus that we're looking at today, you can just see more red. It means the virus has evolved. It's changed from what it was in the Wuhan strain. It's now carrying all these new mutations. And it's continuing to evolve as of course all life does, but you can kind of really visually see it here, that every vertical line is kind of a time point of what we're seeing, and we just see a lot more red in a lot more places. It's a different virus today than it certainly was even 6 months ago. Next slide, please. And the other thing is this is just another slide showing at the different wastewater collection sites with the Miami team and their intrepid field scientists going out collecting. These are all the different capture sites, and you can just see the burst of red popping up, but we do see occasionally individual sites have a slightly different profile. So this highlights the importance of getting more than 1 or 2 sites. If you get multiple sites, you can start to see little pockets of different genetic variation and different strains of the virus that are moving around. So that is kind of fun to see here. Or terrifying, depending on if you're an epidemiologist. Next slide, please. Okay, so what we've done is that was a targeted sequencing where we try to look just at the virus, but we also deployed a method called shotgun metatranscriptomics, which is very much like what it sounds. If you imagine you could take a shotgun and blast apart cells, grab all the fragments of DNA and RNA, and then sequence everything you find, it would give you a broad view of what's present in these samples. It's called metatranscriptome. So the genome is all the DNA of a sample, but since it's an RNA virus and we're looking at RNA, it's a transcriptome, or all the RNA molecules, and meta meaning it's across all species. So next slide, please. So we did that with these samples and had some real questions, like how do we look at the ecology of the wastewater to monitor different kinds of infections, bacteria, viruses, fungi, and understand it broader. And then I think if you click, there's another bullet point there. Yeah, and then so what we did is wanted to look at all these questions, and we basically did a lot of cleaning and quality control first for the data. Then there was called de novo assembly, where we stitched together the fragments of the molecules to make a larger molecule, kind of like a big puzzle, but it is stored in a computer in what's called a De Bruin graph. And then we did a taxonomic classification, basically what species is this, who is this as an organism, and make that mapping, and then look at the abundance and try and classify them. So kind of imagine if you could go out into a field and grab a sample and find all the organisms that are present. That's basically what we're trying to do with the wastewater. And next slide, please. Okay, and there's challenges, of course. Sometimes you have low abundance of things, and we're looking for monkeypox, and it was maybe there at very low levels, for example. And there's always ways to try. We're always looking at ways to improve how we do the sequencing, the assembly, the alignment, the characterization. That part of the noise in the background. My lab is next to a hospital because we're at a hospital, so there's noises. Next slide, please. And then I'll start to show you some of the things that we've seen. So if you look at the bacteria on the left, you can see some of the things that we've been able to pick up by doing this large shotgun sequencing approach. Is you can see over there, really intriguing density of organisms. And so the more red is more abundant and blue is less abundant, but all these things have been picked up, across these different samples. So really each you can see region where it's been sequenced is on top, and you can see the collection from each of the samples at those sites kind of is each vertical line. And then the species on the right just shows what we found. So you can see bacteria, a lot of bacteroides, different species that are present, and that's a very common human commensal. So bacteroides, you'll find vulgaris and uniformis in human feces. It's actually very common. We do see a lot of Pseudomonas also. No, there's an echo, so okay, which also are common, and Previtola are also species we see often in just normal human flora. But you can see there are some that are common, like the Aquaspirillum, which actually is a more aquatic microbe, kind of cool to see. But there is as like the previous point, the very unique profiles to some of the sites, right? So there's probably different things that people are eating, or different. There's suddenly an echo. I don't know if that's me. I think that's me. Do you hear the echo? Or no? It might be. I might just mute for a second. Okay, I think that's
maybe if everybody who's can, just in case, everybody,
okay,
please mute your microphones.
Oh, there we go. There we go. Okay. Okay, great. There we go. Yeah, okay. So but you can see there's just different bursts of color, whereas actually some of the specific pathogens that could be present, like E. coli or Clostridium, you could see some Heliobacter pylori, which is associated with ulcers. You can see even Shigella, even potential evidence of cholera being picked up in some cases, really popping up. And so I think there's some things that are popping up. They're also at low levels, and that's where the confidence starts to drop down. Like Legionella, which causes Legionnaire's disease, is potentially there, or even chlamydia, but it's at low levels. And so to really confirm these, we'd have to validate it in the sample, find the individual patient. But it shows you can see, if it's really just noise, you'd see it just low levels by blue. But we do see these bursts of signal coming from distinct sites, meaning that they could be there just at low levels. And so we're doing ongoing analysis now, but you can see some of the features we've picked up here already. Next slide, please. Then, okay, and then this is just fungi, and also mass versus unmasked. So mass meaning we're cleaning them up a bit to clean up the data. We can see on the left, is a way to actually get a bit more cleanliness. But you can see Melasenia and also some Cryptomonas, various Candida, different fungi that are also very commonly found in environmental samples. These are all also things that are not too shocking to see, but kind of cool. We can get this broad view of the ecology of these samples. Next slide, please. And then, oh yeah, so the other thing we're working on now is really mapping these with other tools and also confirming them. Basically, it's the same way in a lab. If you want to confirm a result, you use a different method. In a computational sense, we can do the same thing. You try multiple methods to confirm what's present and to see what's there. Like, for example, we're seeing bovine herpes virus strains or different phages or rotavirus. You can see over on the right, we can see pepper mild motile virus or tomato viruses. You can see, cucumber viruses, melons, for example. we're kind of picking up also what the population is eating. You can see from these samples, and see the phages, of course, are the viruses that infect bacteria, and they serve as also an interesting source of biology themselves, where they reveal who's there for the bacteria and kind of what people are eating, which some people think it's totally gross to think about, but I think it's pretty fascinating. So you could even see which neighborhoods are eating more fruits and vegetables, which ones are not. So you could even get a population health view through the wastewater, which we haven't done formally yet, but certainly it's possible. Next slide, please. Yeah, and that's it. So it's a quick preview. I didn't know everyone's background in genomics, so I tried to walk people through pretty slowly, but we've, yeah, it's been a really phenomenal collaboration, which actually was an intro. I don't think George is on the call, but we, starting about 2 years ago, saying, hey, well, what if we put together this grant and start to really do a big profiling of wastewater, and got it together in record time. And I sent like 15 versions of documents to Helena, and she managed to still get the grant submitted on time. So, and it's been a really great ride, and more to come, definitely. So that's the data. And I think we go into, I guess, Ken, back to you. Q&A, or do we walk through discussions, or do people send us their stool?
I'm going to provoke you a little bit now. One of the reasons, just to make clear, that this is part of a dialogue in research ethics, because your work goes to the very heart of what is involved in public health and epidemiology is a really important tool, namely surveillance. The discovery of poliovirus in New York reminds us how we would not know these things if it weren't for precisely this kind of surveillance. And what you pointed out, what the two of you pointed out really nicely, is how very many things can be picked up in wastewater. And in fact, one of the things that we've talked about in some of the RADx meetings is whether or not this could be useful for biomarkers for cancer, for example. So as a kind of an early warning system, it can be useful for all sorts of public health purposes. One of the reasons I'm on the grant, and Helena, I was saying to Helena earlier, I do wish they had a picture of me. I don't know how many philosophers find themselves pulling sewage out of the ground, but I'm proud to say that
on the next collection you should go with. You could get, your hands are useful. I think you should go with.
Yeah, usually I generate waste as opposed to analyze it, but intellectually I meant. But the idea is we've got a tool here that is really powerful, really useful, and raises a number of ethical challenges, which is among the reasons I've been invited to participate a little. One of them overarchingly is the role of surveillance in public health. Normally, if you're gathering biological material from people in the hospital, for example, intending to study it, you have to get their permission. But if we were to get permission from everybody whose data or information or biological material were collected in the history of surveillance, it would all grind to a halt within minutes. It is impractical to do that, which is why I think perhaps the most important thing that comes out of RADx is how studies like this rely on the trust of the people that we are trying to serve. And that really matters a great deal, especially in the current context in which I've heard it rumored there are people out there spreading misinformation about COVID. And it's against that background that our scientist colleagues are struggling, because what you're doing is identifying an empirical basis, a reproducible empirical basis that's useful to improve the health of populations. And so the more of that we do, and the more of the understanding that people have, the better. So that's where I wanted our conversation to go first. I mean, when I saw recently where it was a survey of people who during COVID said, "No, I don't believe any of what you all are doing. I'm going to do my own research." You may have heard that phrase. It's become a meme among extremist wackadoos and others who have undermined the nation's COVID response. What you see from this science is that I tried it in my garage, and you can't do it. You need a laboratory that, one, has the resources, two, has the people who know how to do it, and three, the analytic power of all the engines that Cornell and Miami are using. So against that background, let me pause and see if anybody has any questions about that. I want to get a sense of the room about one. I mean, obviously, I am biased and conflicted. I think it's cool and necessary science, and I'm working on a document where I say surveillance is not merely ethically permissible. I think it's obligatory, and that we have that background, I guess, to have a bit of a discussion about. So any comments or questions about that so far?
I guess I'll give the counterpoint for you, Dr. Goodman, and that is, if you find something in the sewage of a community, would that give you the right to then go into that community and go to individual houses to surveil, or go to individual people and say, ooh, we found you have this, or this is in your community and we need to, eradicate whatever is here or whatever? Where do we draw that line on community surveillance and where it impinges on individual surveillance?
Dr. Champney, brilliant question. Thank you for that. It raises the larger issue of our IRB colleagues will be able to identify that perhaps as incidental findings. Namely, you're doing surveillance, but you might find something that could, with fine enough granularity, be clinically useful. And so the question is, what happens if you've got a really strong signal about a malady that, well, our colleagues from the public health department might want to know about? And how would we manage that? And that's among the reasons why there's an ethics component to RADx Rad to be able to address that if it comes up. But Tom, it's a really good question. Chris, Helena, comments?
To me, I think it depends on the severity of the illness too. I know there's a lot of privacy issues and HIPAA issues, but I'm thinking of the extreme of the common cold versus something like Ebola that is very deadly. And so, I think where you draw the line would depend on the risk of death or the severity of the disease as to how far you can go in doing this. But I defer to others.
Who makes that decision about what risk of disease?
Yeah, so it's a great question. There it is. It's like most questions that are going to be public health. Should involve feedback from the community, from public health experts, from politicians. But what's interesting is people will still, I think, even just disregard them sometimes, but at least it has to engage the community when you make the decision. And there's other examples too. For example, there's pilots being discussed. We were talking about doing wastewater surveillance from airplanes. What if you could sequence what's in the wastewater on the way to a location? And then if there's a positive, who makes the call to quarantine everyone that's sitting on the plane? I can tell you, I don't want to make that call. I can work on the sequencing and the informatics and what it is, but someone's going to have to, though, basically, be the final call and have the buck stop with that person to say, I've made the call, yes or no, on the risk. And that person's going to have to take the heat. Now, normally it's politicians, right? So my brother's a mayor of a city in Wisconsin where I'm from, and the pandemic has been a really hard time for all politicians and public health experts because they get all the blame and all the heat, but really no credit when things go well, which is kind of par for the course for any politician. But it's been exceptionally bad recently. So I think where it likely is to go is to, someone who's the head of the public health office for a city, and they say there's going to be advice and there's going to be community and feedback. But at some point, someone's got to just make the call and say, this is too dangerous. We need people to, sometimes it could be simple. Telling people just to stay home for a little while is not so bad. I think also we've all gotten used to that. But that's easy for some people to say if they don't have to go to work for their job and they're in a lower income house, they have to go to work or they'll lose their job, right? So I think there's, it's easy to say, oh, just stay home, but it's really not possible for a lot of people, or actually most people. And so I think all those things have to be factors. I'm just kind of blathering now, but all those things are factors.
One of the things that came up during the course of this was the team's effort to make sure the community did know about the research. And so it still goes to Dr. Champney's point that ultimately becomes a question you can argue of health literacy. Do the people we're purporting to help keep up on the news about this sort of thing? And so there have been newspaper articles internally in UM's media. There have been articles about the science here, and ultimately disclosing that in fact this is occurring. What makes this credible at the outset is this is the way we've done surveillance for a long time. Foodborne maladies, for example, if you go to a restaurant, you hope the Department of Health has been inspecting that place, and they may find people who got sick. We have questions of contact tracing that arise all over health interventions that require sometimes some very awkward conversations. Our colleagues in public health and the health department deal all the time with reportable maladies. If someone shows up with a reportable malady, sometimes that will trigger a contact tracing visit. And we are able to do that Maybe reach out to people who didn't know they'd been exposed to let them know, because that's the way public health works. And I think that's an important baseline. But it also means, Tom, that what you've done is said that having a process to make those tough calls is really important. And that's among the things that is partly my responsibility as part of the project. What would happen if we were to do that? We would in fact communicate with our colleagues in government. I shout out to colleagues in government again. I'm sure if you work for the government, one, you know you're appreciated, and two, you don't have near enough resources to do what you'd like to do. Imagine something like RADx, not just in Miami, but in Orlando, Tallahassee, and Jacksonville. If we could do that with the appropriate structure, we could learn a lot about how to improve and protect the health of populations. George, I see you've materialized on my screen.
Can I push
just
back
wanted
just
to
one
comment.
more bit? And that is, should you limit what you're looking for in the wastewater? It seems to me that we're doing a wide expanse, and you may be telling people, oh, we're looking for SARS in the wastewater, but you're really looking at all sorts of other things in the wastewater as well. And it stretches to me, you know, if you start measuring HIV in the wastewater, are the communities going to want to know how much HIV is in their community, or other things that might be ethically challenging? Should there be a limit to what you can or cannot measure, or should someone be deciding what can or cannot be measured in all of that realm?
I think that becomes a very difficult question, because you know, you really do want to look at things that are off target so that you can determine unexpected surges. You know, certainly we never expected this particular pandemic. So if we had just kept blinders on just for influenza, we would have never actually seen COVID coming up. But I think also in terms of, you know, what do you do with the information? It's important also to consider the scale and scope of the surveillance. The information that we've generated with this project has helped inform public health policy decision makers, both at the university level and at the Miami-Dade County level. And what they can do with that, and what they have done with that information, and what they should do with that information is very different depending upon the scale and scope of the community that you're surveilling. So, and of course, it becomes a very different question when you talk about regional, you know, statewide or nationwide types of decisions.
Really good point, George. Thank you for that. Mind you, one of the things, this is a comparatively young project. The NIH was keen to do this, but we won't. Helena, how long has it been going on? A year and a half now, or 2 years?
2 years. 2 years.
So part of it, and this is what are we learning from this that will help answer these questions. There's a website, I mean, that engaged citizens can find out more. What could or should we be doing to engage more against the background of all the other kinds of surveillance that are occurring, right? Which is why trust looms so hard. Most of us sort of muddle through our days thinking, I'm counting on the health department to keep up on this for me, so I don't have to do it. That's what we count on in civil society. But a recommendation, Tom, that might be useful when this is done is a community advisory board, for example. You know, a number of newspapers around the world have written about wastewater surveillance, and ultimately it requires an engaged citizenry and an accurate and free news media to do that. You know, could we send letters to everybody in town? Could that Dade County Health Department send have a newsletter that goes to everybody? It says, here are all the ways we are trying to keep you safe. If you have questions, let's talk about them. Because the point, of course, is that this is not the unintended consequences that you've identified, Tom, are really important. But Chris's response, imagine we could have detected Ebola and saved a bunch of lives. Right now, we're not testing for it. Let me ask you, Chris and Helena, how is it decided which particular organisms to test for?
Well, we started with SARS-CoV-2, the virus that causes COVID-19 because of the pandemic. And so that's one of the questions that we have is, you know, what do you measure and how do you move when you move forward? What is it that you measure? And I think the shotgun approach that Chris mentioned is a great way to start identifying. I mean, the pathogen list on the right-hand side was amazing. And an important aspect is, because you see it, you need to, is there, you know, what's going on? What does it mean? And so we need to start establishing baselines. That was another comment that we provided to the NIH as far as future work. You know, what is a normal baseline versus what is above normal? So
one of the things
I want to add something to Tom's comment. The other extreme of surveillance and why this all, how did this all start? Who even thought about using wastewater? This all started, and we're actually funded through an NIH Institute on Drug Abuse, National Institutes on Drug Abuse. And so this all initiated this idea based on actually enforcement, people trying to, law enforcement trying to use chemicals, looking at the chemicals in the wastewater to identify drug laboratories, illegal drug laboratories. And so it was used for that level of enforcement. But that's a different purpose altogether. But you know, at what point do you cross the line? So I just wanted to throw that out there, that this originally started through the law enforcement community and the technology that they were developing for tracking chemical contaminants.
Which can be scary. I mean, let's take the example of Russia. Would you like the Russians examining all the wastewater of all the people in Russia and determining, you know, we live in a freer society, but one worries about other societies and ways that even our society may abuse that technology.
Yeah. I mean, I don't know if people know this, but Putin is famous for not leaving any stool behind. He has it collected actually, because he's worried that someone will scan it, find a source of a disease or a virus. I tried to get my wife to do that. She declined for myself, but you know, it's a personal choice for sure. But yeah, you know, there's national security implications. There's privacy implications. These things are actually not even hypothetical. It's happening.
So against that, I mean, so one of the points, Helena, that you raised in response to Tom's question is, this is to look at maladies that spread in populations, and therefore it would be out of bounds if someone were to say, we'd like to start looking for metabolites of cocaine in dormitory wastewater, right? Or whatever. That would be a sort of thing we'd say no to, right? Unless, of course, we had a separate study that was intended to look for that, and that might change the entire profile of how it's communicated to the people who are purporting to serve. Is that a fair statement?
Yeah, I think the chemical contaminants and the drug use is a different question altogether and for a different purpose.
I think one of the things we've gotten feedback from the public health decision policymakers, particularly at the university, is if we suggest looking for a certain new target, that what they really want to know is what's the result if we find it. You know, I mean, one of the nice things about wastewater is that you don't have to actually identify individuals, and they don't have to have the onus of doing their own clinical testing. So it's sort of like an unbiased type of approach in that sense. You know, particularly if you're looking at a broad region where you can't identify a specific group, you know, for that. But even with that, you know, you can see the prevalence of something, but if you can't do anything once you see it, then the question is, why are you looking at it? You know, if you can't do anything about it. So that's been something we've been asked to you know, consider in determining what are the new targets we should be looking at.
It's a great question for scientific methodology too. I mean, this is not exactly hypothesis testing, right? We don't even know what we're looking for to frame future hypotheses. And so, George, what this suggests, what your point suggests is, you don't get what you ask for. On the other hand, if you never asked, you wouldn't know how to frame questions for the future. And that's been the history of surveillance for a long time. Remember, polio was detected in wastewater many years ago. And being able to know what communities are at greatest risk is a very powerful tool for our colleagues in public health to be able to figure out where to have an intervention or an educational intervention, I guess, although I don't like that phrase. More comments and questions? Fortunately, this is not Russia. Although I'm sure Vladimir Putin would love to be able to tell people of Florida how to live, they don't do that yet. But this is why the checks and balances that are in place in civil society are in civil society. The way to prevent anything from going wrong is never to do anything. All right, and the burden to the health of populations from that stance would be too much. Also, if the granularity of wastewater from a shared sewage disposal or transmission system is just not there to identify individuals, right? Could it be done in principle?
As I understand it, the Japanese have toilets that are designed to do that, to examine your stool as it passes through that toilet to again then give you a personalized health check.
For free? It's worth. Go, Chris.
Yeah, there are some that like capture it and look for signs of disease. There's some, one of my favorite ones is actually can tell as a smart toilet that actually can examine when you sit down who's sitting down, and then it'll change the temperature based on your preferences. And so, they call it like I'll put it in the chat box, but there's a really funny article about this, and they called it, it's like the fingerprint, but it's based on what you can see when you sit down. And so there's a really kind of a hilarious article there on from Business Insider. But there's all this information, so you can tell who's going where, when they're going, who it is. Of course, there's human DNA, as I alluded to as well, that's there. So you can actually, if you're grabbing from an individual toilet, you can sequence their genome, right? And that has implications for genetic information and non-discrimination. There was an act called GINA that was passed to prevent this, but actually that only applies to when you ask someone for a sample. If someone leaves it behind, then you're not violating the law, actually. So it's an interesting deviation there. And actually, we have someone coming to the meeting, the genomics meeting that's going to be at Miami in November 19th and 20th. One of the speakers on one of those days is talking about how he narrowed it down, an infection down to a single toilet in Wisconsin. So we'll see a case report of that kind of at the meeting in November.
A single toilet in Wisconsin sounds like it'd be a great title for something.
Yeah, I think it's a Kenny Rogers song, maybe I don't know what it would be.
Elaine, before you ask your question or make your comment, the origins of this in politics go back a long ways. When foreign visitors go to the White House, for example, or the Kremlin or Versailles or wherever, years and years ago, they had traps that would divert the contents of a toilet, so intelligence agencies could examine the waste from heads of state to try and predict their longevity and any maladies they might have. Which, if it's the head, of some other country that you're having an issue with, helps you do long-term planning for the government. Dr. Sternberg.
Thank you for that. I think that the point that you made about where you collect the waste sample will be relevant. I mean, you mentioned, for example, that some of these were dorm specific. And so depending on where you dipped your collection can, you might be able to know to locate it quite specifically without the intelligent toilets. But against that, you have the idea that this is a captive audience. These are people who have chosen to be associated and to live in a dorm, and therefore they can be required to give consent or simply absent themselves. It's much harder for the population overall. But against that as well, I would suggest that the questions that are being raised here by the advent of the technology are ones which recur at every level. You pointed out this has been done with visiting dignitaries. The question about surveillance, the questions Tom mentioned are very general ones, which we have to consider, and they're not new. And so to the extent that methods have been employed for addressing these questions, these have to be used as well. None of the questions are new, none of them. I'll stop there.
Every once in a while when I teach this course, I show, if you haven't seen it, look at John Snow's map of London, where without any consent whatsoever, he put dots on maps where people lived to be able to try and track the cholera outbreak. It's a great, so thank you for that, Elaine. It's a great point about, we've been struggling this for a very, very long time. Where doing nothing is arguably indefensible in terms of public health. but what we do needs to be guided, and which is why the 3 cheers to Chris and Helena for and the rest of the team for making ethics part of RADx. Right? Did you say at the NIH meeting, Helena, what are the RAD sites are doing ethics?
We're the only ones, and so and then we were commended for that by our NIH program managers, that we were unique in that sense. So thank you.
The ethics, and some of the technical methods for sequencing. Even in early days, we did a bit of LAMP, new different methods for amplification. So yeah, I think we've been, and also had multiple papers come from the work, multiple other ones in the works, and I think we have there's a dashboard, there's a website, and I feel like there's a lot of resources that have come from the project, which have been great.
If anything, I would suggest that all these types of wastewater projects should be mandated to have a bioethics component, similar to what they did during the Human Genome Project, where all of that funding was, any proposal had to have that type of component in order to actually receive any funding. I think, as we move forward in this field, and we're really in the infancy stage of it, that's something to consider in terms of where we might go to help assure that these types of issues are addressed.
It's a really good point, George. In fact, I mean, one of the things that some of us have so far tried unsuccessfully to advocate is that if you're doing informatics, and it could be clinical informatics or bioinformatics or public health informatics, the more lives are touched by these machines than they are by genome science. And so while you're right, George, while the Human Genome Program created ELSI, the Ethical, Legal, and Social Implications Program, that has not been repeated anywhere else. And some of the work that's done in informatics, including bioinformatics, is really quite interesting in this regard. To say there's an ethical issue is not a criticism. It's to say that we are mindful of this and we want to get it right. The point of ethics is not to say, stop, don't do that, run away. It's to say, here's how. And that, I think, is why your program officers are so excited. There's nothing that'll warm your heart more than a program officer saying something nice to you. Wayne, is your hand still up, or is that a new question or an archival question? All right.
Apologies.
No worries. Any other comments and questions? We're coming on to the hour, and I know people have calendars that are merciless. And so I'm going to count to 3. That was 4. And then ask our presenters to give some closing remarks. Just courtesy of the visitor, Chris, talk big for a second about next steps.
Yeah, I think this is the tip of the iceberg. I know it raises a lot of big questions, but I think as we started to get to in the Q&A, there's more and more democratization of access to these technologies. So I think a lot of people will see a lot more go into the homes. People already see home testing for COVID has skyrocketed, and it means people don't report things as much. And the good thing about wastewater is even if they don't report it, at some point everyone's got to go to the bathroom. And so the importance of this is that it lets us measure things even when people start to keep things in their own home. And it could get more interesting in the future, where you set up a little sequencer in every toilet and someone could get a report. I mean, I think the scary part could be it could be used for surveillance in a nefarious way, but the exciting part could be if you went to the bathroom at night and there was an analysis happening while you were sleeping, and you got a report in the morning of your microbiome health, any diagnostics, and you could get it on a daily basis. Maybe you wouldn't need it every day, but you could push a special button on your toilet to get a deeper analysis. I think it'd be fabulous. Of course, it's a bit expensive right now, but in the future, it would come down in cost, and I think enable earlier tracking of diseases and maybe even prevent outbreaks before they become really pandemics in a dream world. So that's what I'm hoping for.
Great point. Thank you for that. Helena, last word.
Yeah, no, I think that this conversation was excellent, and the ethical issues to me are fascinating. Going into this project, I never thought of the ethical issues in terms of having to report, not reporting, what to report, how to report. And maybe in the next round, it would be good to be able to address some of these questions, for example, that were brought up in terms of how far is acceptable, can it be related to the risk of the illness, the severity of the illness, the size of the population that's being surveilled. So for example, is a dorm room too small, too big? So I would imagine at the wastewater treatment plant where you have 800,000 people contributing, that's more anonymized versus a small, as the buildings get more and more specific. So at what point is it acceptable? And I wish we could answer these ahead of time, and maybe for the next pandemic, we would be ready to do that. I also looked up Havasupai. I saw that in the chat box. I thought that situation was fascinating. And I thought even in the case of the diabetes 2 study with the Havasupai tribe, that, you know, it's a homogeneous population. And so even if the population is very big, I think we need to take into account how that the results would be maybe generalized to that population, which may not be desirable. So there's much more to it than maybe my simplified original view of it. This is very complex, and I would love to have a system in place ahead of time, already well thought out as to, you know, when can we report? When should we report? When do we have to report? Kind of answer. So,
great point. The Havasupai, I recall, are an indigenous American tribe in Arizona. They're working with scientists there and agreed to have some genomic analyses done for metabolic disorder and diabetes. And then found out the scientists looked at a bunch of other stuff, including issues in behavioral genomics. And it became a really good example of how trust in a community can be easily undermined. So we're having this discussion as a way to support RADx and our colleagues who are doing this to ensure that the trust continues to be deserved. Against that background, thank you so very much, Chris and Helena. On behalf of the Institute for Bioethics in our 31st year, you guys have done a great job in leading us off on a program that I think has come to be one that we all enjoy, in part because of colleagues like you. Thank you very much. We're adjourned.
Pleasure.
thank you, Ken. Thank you so much for this
Thanks.
opportunity.
Thanks.
Bye.